Welcome students. Today we are going to discuss chemotaxonomy. Chemotaxonomy has a great antiquity and the chemical characteristics of plants have been noted and used by taxonomists. Its early history is largely a history of use of plants in medicine. The herbalists of past began to group plants possessing similar medicinal properties. Grashop, 1909, published the presence of tannins, alkaloids, cyanogenetic compounds, and saponins in a wide variety of plants. He suggested the use of comparative chemistry in taxonomy. McNair, in 1935, has tried to apply comparative chemistry in taxonomy. According to him, any one alkaloid rarely occurs in more than one family, but may occur in several members of one family. For example, protopine in the Papaver family. This alkaloid, protopine, is not found in any plant of other families. McNair, 1935, has discussed angiosperm phylogeny on a chemical basis and has tried to use alkaloids, glycerides, for example, fats and oils, volatile oils as aids to taxonomy. According to him, plants can be classified chemically in accordance with the substances made by them. Such chemical classification may be compared with or used as supplement to morphological classification and may be of some importance in the development of the true natural system of angiosperm phylogeny. He further adds that the chemical products of more highly evolved plants have larger molecules and that the iodine number of glycerides is higher in the more highly evolved groups. According to Hutchinson, Fumarolidae is quite distinctly separated as a group from Papaveraceae proper and closely allied to certain genera of the family Berribidaceae, for example, Epimedium, Aceranthus, and Bongadia, and there is close affinity with certain genera of Papaveraceae, especially Chelidonium. He writes that this alliance is more apparent than actual and that the Fumaraceae have not arisen directly from the ancestors of the present Papaveraceae. Now, classification of chemical characters used in taxonomy. There is no suitable classification of chemical characters used in taxonomy. In 1966, three principal kinds of chemical constituents were recognized which help in taxonomy. Number one, primary to basic constituents like proteins, nucleic acids, derivative chlorophylls, and polysaccharides. Number two, secondary constituents which lack nitrogen and are not involved in the basic metabolism of cells. Number three, miscellaneous substances. These chemical constituents are classified according to their molecular size into following. First, micromolecules. Compounds of relatively low molecular weight, for example, alkaloid, amino acids, cyanogenic glucosides, glucosinolates, pigments like anthocyanins, betalains, and so on. Phenolics like flavonoids and so on, and terpenoids. Second, macromolecules. Compounds of high molecular weight such as proteins, DNA, RNA, cytochrome C, ferredoxin, complex polysaccharides. Number 3, proteins. Micromolecular data have practical utility in taxonomic problems at the generic level or below. They have been used to study variation caused by natural hybridization as well as other problems. In such studies, micromolecular data have been treated as characters used in classification. Generally, macromolecular data are most helpful when applied to problems of phylogeny above the generic level. The taxonomic important chemical compounds and their systemic value is as follows. Number 1, proteins. Proteins serve as most important tool in chemotaxonomy. The importance of proteins in chemotaxonomy has several reasons behind it. For example, first, they are large, complex molecules showing little qualitative variation with changing environmental factors. Second, they are universally distributed. Third, they are relatively simple to extract and present in appreciable amounts. Fourth, cheap, simple and rapid methods have developed in recent past for protein analysis and comparison by taxonomists, of which electrophoresis is particularly an important method of protein separation. The variation in protein structures between plants effectively provides an inner view into the cell's genome, as the amino acid sequences of proteins are encoded by the nucleotide sequences of a cell's DNA. Proteins have been used in taxonomic investigations in the following ways. Number first, comparison of the protein binding patterns. In recent times, protein binding patterns obtained by gel electrophoresis have been focused on the problem of identification of critical taxa, their relationship and taxonomic status. Taxonomic interpretation should be based on comparison of proteins from homologous organs of the same age in order to avoid any confusion due to variability of proteins. Considerable variation in protein complements has been recorded at the level of species and genus and even between the same plants in different populations. These form evidences upon which taxonomic systems may be founded, tested or demolished. It has been possible to obtain useful systematic information from electrophoretic analysis of crude biological protein samples. For example, Johnson and Hall 1965 have demonstrated the phylogenetic affinity in Triticini by the process of protein electrophoresis. This process also helped in establishing a close relationship between Vicia and Letharis. Interspecific variation among 8 species of Cassia were evaluated on the basis of these protein and mitochondrial DNA RFLP by gel electrophoresis as well as pollen protein patterns. Number 2, amino acid sequence studies of homologous proteins. Comparison of amino acid sequences of homologous proteins from different taxa has also provided a powerful tool for evolutionary and systematic studies. The degree of similarity in the amino acid sequence is presumably proportional to the degree of genetic relationship. For example, based on amino acid sequence studies, Martin and his associates have tried to trace the phylogeny of various taxa like Malvaceae, Ranunculaceae, Magnoliaceae, Polygonaceae, Martales, and some Solanaceae, and have elucidated the phylogeny and taxonomy of angiosperms. Number 2, amino acids. There are 2 groups of amino acids present in the plant cells. They are: first, protein amino acids; second, non-protein amino acids. Now, we will discuss them one by one. Number first, protein amino acids. They are the building blocks of the proteins and are raised on hydrolysis of proteins. They are universally distributed in plant tissues. Amino acids are present in the free form in various plant cells. Has also been used as a taxonomic factor in some cases. Shallad and Joliffe 1968 studied the free amino acid composition of the pollens of 11 different grass species and found it to be similar in all the species. Generally, free amino acids are usually higher in pollen than in leaves or other plant tissues. Hence, free amino acid composition of pollens has been used to study the homology between various species. For example, Asteraceae, Cassia, etc. However, it is very difficult to draw any conclusion on evolution based upon the data on free amino acid composition alone, as amino acid composition alone greatly varies with climatic and nutritional conditions as well as with storage and handling patterns. Second, non-protein amino acids. They are not found in combination in proteins and are more numerous, estimated to be about 300 in number. The non-protein amino acids are discontinuous in distribution and less susceptible to rapid change, which increase their taxonomic value. They occur in plants which are only distantly related. The occurrence of such compounds may be explained by the fact that different plants can probably produce the same amino acid by different biochemical routes. Several workers have studied the taxonomic potential of such amino acids. Following are a few examples of taxonomic significance of specific non-protein amino acids present in different groups. First, acyl ornithine has been identified as the main non-protein amino acid in Fumariidae under the Fumaraceae. It has also been found in ferns and grasses. Second, cannabinine have been detected only in 60% of the species under Lotoidea of the Leguminaceae. The prevalence of cannabinine was considered as an advanced character as it was unknown from the primitive tribes Podalirae and Sophorae. However, this amino acid was subsequently discovered in both the tribes. Third, the amino acid azitidine 2-carboxylic acid is extremely restricted in its distribution, being present in the Agavaceae, Amaryllidaceae, and Liliaceae. The amino acid is also reported from the legumes Dillonyx and Peltophorum. Fourth, Lathyrus species could be grouped under 7 infrageneric groups on the basis of association of amino acids within the seeds. Each group is characterized by a different amino acid or group of amino acids. Fifth, species of the section Gummiferae of the genus Acacia can be recognized easily on the basis of amino acid contents of their seeds. Sixth, 4 infrageneric groups have been recognized in the genus Vicia on the basis of distribution of amino acids. Seventh, free amino acid lathyrine is also known only in the genus Lathyrus, and this supports the present circumscription of the genus. Now, we will discuss flavonoids. Flavonoids are the largest group of naturally occurring phenols. Phenols contain the hydroxyl group attached directly to the aromatic nucleus. They are mostly found in the vacuoles of the plant cell. Various types of flavonoids include flavones, flavonones, isoflavones, isoflavonoids, flavonols, anthocyanidins, chalcones, aerones, and biflavonils. Cronquist and Takhtajan placed the jugulandales in the Hemimelidae, but Thron assigned Jugulandaceae to the rutales. But the presence of 5 methoxylated flavonols relationships with the Hemimelidae. Flavonoids are present in leaves, flowers, and fruits. They function not only as insect repellents but also play a vital role in resisting chronic pests and pathogens, and by interaction with plant hormones, they regulate certain growth processes. These chemically complex compounds are easily and quickly identified, have great structural variations, widespread in distribution, and physiologically relatively stable, which makes them taxonomically significant. The use of flavonoids in evaluating contemporary classificatory systems has been mainly based on their distribution patterns, that is, their presence or absence. Phytochemical taxonomists have considered them as evolutionary markers due to many positive correlations than they display. The primitive and advanced types of flavonoids in angiosperms were deduced by Harborne in 1977. According to Swain, 1975, during the course of evolution, flavonoids increased in their structural complexity from the primitive glycoflavones in Cara to the proanthocyanidins in ferns, gymnosperms, and putative angiosperm progenitors. The content of flavonoids have been found to vary in different plant organs and have also been found to vary in response to light and growth. The flower pigments, which are usually anthocyanins and anthoxanthins, vary greatly and have been shown to be under genetic control. It is also reported that the anthocyanin production increases with the deficiency in nutrients such as nitrogen and phosphorus. However, all variation in pigment production is not genetic in origin. In general, vegetative tissue extracts are more reliable and convenient in chemotaxonomical evidences than flower pigments, which are too variable. Many phylogenetic inferences have been based on these characters in various groups. Following are few examples. The flavonoid systematic of the genus Perideridaria was studied by Giannesi and Chuang in 1976, who found that although the 16 species studied by them had the same flavonoids, they could be categorized into 3 discrete groups. They are: first, those producing only flavonols; second, those with flavonols mainly and a few flavones; third, those which produce flavones predominantly. An exclusively survey of the families Umbelliferae and Arelaceae by Hagnier in 1971 revealed that the 2 families are closely related. Hagnier also concluded that they may be regarded as constituting the order Urabiliales, which in turn may have evolved from a Rutilian stock. Analysis of flavonoids of leaf of Liliaceae Juncaceae, Cyperaceae, and Poaceae suggest that all these families have arisen from liliaceous ancestors. The flavonoid pattern of 5 species of Tragopogon of Asteraceae and their F1 and F2 hybrids were studied, and all the species and their population showed different chromatographical results. Flavonoids may have been used in the estimation of generic affinities in Ulmaceae and of species relationships in Chenopodium. The evolutionary pathway in many taxa, for example Lemniaceae and Ruellia, have been traced based on the data of flavonoids. Flavonoid chemistry has been used to distinguish species of Spirodela which cannot be distinguished on the basis of their morphology. Now, number 4, terpenoids. The order of flowers, fruits, leaves, for example, mint and eucalyptus, and wood, for example, sandalwood, is due to essential oils, natural organic compounds called terpenes. The terpenoids have been classified on the basis of number of isoprene units present in them. For example, monoterpenoids with 2 isoprene units, for example, chitral, limonene. Sesquiterpenoids with 3 isoprene units, for example, farnesol. Diterpenoids with 4 isoprene units, for example, vitamin A. Triterpenoids with 6 isoprene units, for example, squalene. Polyterpenoids with several isoprene units, for example, rubber. Some of the chemotaxonomic applications of terpenes are mentioned below. Sesquiterpene lactones are common in the family Asteraceae, in which the oxidation level of these sesquiterpenes is sometimes specific to a tribe, subtribe, or even a genus. The absence of sesquiterpenes in the Asteraceae may be rare. It has been found that if ambrosia and its allies with Francesia and xanthium are removed from the helianthie, then the residual elements are similar to the helianae in terms of lactone distribution. Thus, there is a need to construct a separate tribe or a distinct family to accommodate allied forms of ambrosia which exhibit diversity in lactone production. Second, geographical races of Pseudotsuga menziaceae were distinguished based on the terpenoids of their conical oleoresins. Third, the classification of eucalyptus has been difficult on the basis of gross morphology. Later, the genus was divided into larger groups containing different oil combinations, which they correlated with leaf venation and bark structure. Fourth, origin of certain citrus cultivars was determined by studying their rind and leaf terpenoid pattern. Fifth, a survey of monoterpenes in 19 species of salvia indicates that terpene composition is a valid morphological trait in the analysis of introgression within a group. Sixth, gum terpentines have been used in the taxonomy of pines. Seventh, there are 3 sympatric taxa of Hedioma coexisting without any sign of natural hybridization and used their terpenoids in identifying them. Now we will discuss alkaloids. Alkaloids are a heterogeneous group of organic nitrogen containing bases, often with a heterocyclic ring. The true alkaloid have a nitrogen containing heterocyclic nucleus derived from biogenetic amine, and they can be related structurally to parent bases such as isoquinoline, pyridine, piperidine, and tropane. They are byproducts of plant metabolism and are synthesized from amino acids and their derivatives. Three main categories of alkaloids are distinguished. Number 1, true alkaloids. They contain a nitrogen containing heterocyclic nucleus derived from a biogenetic amine. Number 2, protoalkaloids. They are derived from amino acids but lack any heterocyclic ring. Number 3, pseudoalkaloids. They are biogenetically unrelated to amino acids and are derived from terpenes, sterols, aliphatic acids, nicotinic acids, or purines. Alkaloid content can be considered as a source of taxonomic evidence as alkaloids characterizing species of a particular taxon or frequently of the same chemical or biogenetic group. This suggests that related plants share the same pathways of alkaloid synthesis. Therefore, alkaloid content can be considered as a source of taxonomic evidence. However, mere accumulation of alkaloid does not imply taxonomic relationship at family level. Some of the taxonomic aspects of alkaloids are mentioned below. Number 1. Distribution of alkaloid has proved useful in the taxonomy of the Fabaceae. Of the three genera viz. Genista, Amodendron, and Adenocarpus, under this family, Genista and Adenocarpus were included in the tribe Genistae, whereas Amodendron was placed in the tribe Sophari, which is characterized by the presence of matrine alkaloids. However, phytochemical studies have shown that all the three genera contain amodendrine histidine alkaloids, which suggest that amodendron should be transferred to the tribe Gentaceae. Number 2. Lupin alkaloids have been found in three tribes under the subfamily Lotoidea of the Fabaceae, viz. Sophari, Genistae, and Podalari, which suggests that these tribes may have originated from a common ancestral stock. Number 3. Alkaloids are useful in taxonomic studies in papaver and argemone, veratum, as well as in Lycopodium, lupinus, and Caryophyllus. Number 4. The presence of isoquinoline alkaloid in the families Fumaraceae and Papaveraceae indicates very close relationship between the two families. Fifth, on the basis of the distribution of benzyl isoquinoline alkaloids, several modern taxonomists including Takhtajan, Cronquist, Thron, and Dahlgren rearranged the families with Apocarpus gynoecium in Magnolidae. Dear students, this is all about chemotaxonomy. Thank you very much.